Hi everyone, welcome back to the channel. Dr. Asif explains. Today's topic is BSC 1005 Module 3 review. So this Module 3 has actually three chapters in it. They are 10, 11, and 12. Chapter 10 is DNA technology. Chapter 11 is evolution of populations. And chapter 12 is macroevolution. So let's get started. This is for the upcoming exam. So as always, start with your PowerPoint and then do some deep reading from your um, textbook, Concepts of Biology, the Orange Book, and read chapters 10, 11, and 12. So what are some key points? So if we go over some key points, chapter 10 is about DNA technology. We have six major DNA technology actually. So pay attention to the six major topics here, right? Under chapter 10. So this is chapter 10, DNA technology. This is also called biotechnology in older term, but same idea. So if we just look at the PowerPoint, DNA technology, there are a lot of new techniques coming out. Some of these are very, very new. Some of them has been around pretty long time, like genomics, bioinformatics, and some of them are getting updates. So always look at the vocabulary. So for example, genetic engineering, it's been around maybe 30 to 40 years. So you should pay attention what you look at this vocabulary here and this in your PowerPoints for the enzymes, DNAs, DNA ligase, DNA cloning, fragments, DNA probe, vectors, plasmid splicing. There's some vocabulary in the chapter 10 for DNA technology, PCR, right? Polymerase chain reaction, and some other techniques. Um, so what is DNA technology? It's actually a molecular technique or a number of techniques. Basically, it could be used for many different things. Like you see here, DNA profiling, for example, one example. So to establish guilt or innocence, right? So DNA profiling, which has been around for a while. So so cells of the person so cells of the individual people have a unique DNA except identical twins. So based on this theory, we can use DNA technology and profiling technique to establish guilt or innocence, compare control with crime scene and suspect number one, suspect number two, suspect number three, and then see DNA match, right? So another area is genetic engineering in plants, crop plants. This is pretty common. So do you see, you hear every day GMO plants, and you go stores, you see genetic GMO tomatoes or GMO corn. So what is that? That's actually genetic engineering. So those tomatoes are genetically engineered. They may have an additional new gene from outside, cut and join using restriction enzymes and ligase, and basically, or E. coli and some other system, basically gene inserted into the plant, and a new product is a GMO tomato, for example. So GMO organisms acquire one or more than one genes artificially. And then we call them also transgenic organisms, which means if a gene come from another species, then it will be a transgenic. Okay, so pay attention to these. Biotechnology is the overall technique. Talking about GMO, transgenic. So look at how actually bacteria use, since actually we know from the earlier chapters, bacteria is a good system actually for the use in gene cloning. Okay, so look at these some details. Gene editing is a new technique, maybe last 10 years, already awarded Nobel Prize in Physiology and Medicine for this technique, even though one of the newest but most very exciting technique. This is also called CRISPR-Cas9. because of the enzymes used in this gene editing. It's called CRISPR system. So what's so cool about it? It's actually working in living cells and correcting maybe a gene mutation in a living cell. That makes it really, really exciting. So using CRISPR-Cas9 genetic scissors. So the cutting with the Cas9 and repairing using some repairing enzymes, and then let the mutation actually knock out, maybe disable a specific gene. Also, you can knock out again a disabled gene or mutation, possibly alter, edit a living cell. That's gene editing, right? There are a lot of medical applications, like you see here in this humulin, which is a human insulin, which is actually made in a GMO bacteria, produced by a GMO bacteria, which is a very good thing. So GMO, we see a lot of examples, golden rice, or golden rice 2. In this case, you see the golden rice on the right side here, which is golden color, which is a GMO rice, which has additional genes, including to make extra vitamin A in it. The cassava you see on the second picture here is also added beta carotene to increase their vitamin A levels. And so in cassava. Human gene therapy is, or gene therapy in short, this is also a technique, a DNA technique, a rather very selective technique that's actually also use maybe a normal gene inserted to human to treat a specific disease. So this is well, it's in a clinical trial. So this technique, unlike a lot of other techniques, it's very, very selective and technique. DNA profiling, I already talked about. So PCR is one common technique in DNA technology. It's used in so many areas. And you can see here, if you have just one small, tiny piece of DNA, you can actually make millions copies of it using actually PCR machine. Bioinformatics is another DNA technology, which is also called well, one area in bioinformatics is also called DNA sequencing. So there are newer and faster and cheaper DNA sequencing techniques coming out all the time. So like next-gen sequencing, you can see here is our third-generation sequencing, for example. So the idea is complete nucleotide sequence of a DNA of a specific organisms. Okay. Genomics is been around a long time. As you see here, so many organisms, we already have complete genomes already we know. And there is a little information here about this Y chromosome, and Y chromosome have specific genes, but one unique thing about Y chromosome is it's actually it goes from father to the son intact, and that actually helped some studies like you see here, 8% of the males in Central Asia may be descendant from Genghis Khan. How we know that? Because of Y chromosome. So there is a summary of this whole chapter 10. Also here, also in your slides, in Canvas, and in your study guide. That's chapter 10. Chapter 11 and chapter 12, they go together. They are both about evolutionary biology. So chapter 11 is evolution of populations. Chapter 12 is macroevolution. So a couple of things about chapter 11. So this is basic evolution and evidence and evolutionary evidence and mechanisms and techniques that actually cover here. So some vocabulary, carbon dating, Charles Darwin, cladograms, biological evolution. So you might want to actually look up and study. So so what is evolution of populations? First, only populations evolve. So that means changing next generation's populations over time. Individuals, not really, but they can mutate, or they can be naturally or just selected, basically. They do not evolve during their lifetime, individuals. So it's only populations. That's number one. Rule number two, most recent common ancestor idea is actually same as it means closest evolutionary relationship. And number three, any two species under same genus they are more closely related. So always remember that. So we know from the chapter 1, this whole diversity of life and taxonomy and genus and specific epithet. Every living species, like 1.8 million species, have their scientific name, which uses a binomial system. So, and pay attention to the genus names I just mentioned. Species under same genus, they are closely or more closely related. So, evidence for evolution, we have, of course, a lot of fossil records, and so pay attention to fossil records, and then Darwin's book, Origin of Species, and he's actually so his theory, basically natural selection as an evolutionary mechanism, so differential reproduction or survival. And then he talks about biogeography, right? So if if uh, if if two species are in a nearby island or an island nearby, uh, one is an island, the other one is a nearby mainland, they are actually they are the species live closer, they tend to be more closely related. That's biogeography. And natural selection is about more about how the species adapt to this changing environment, and talks about that. And that's natural selection. So there is more. There is his observations from Galapagos Islands about biogeography and adaptations, and then his theory, right? So of basically natural selection, adaptive evolutions of populations to fit environment, and we have fossil evidence. Even though some of them are incomplete, but we have fossil evidence, and then currently they are also searching more fossil evidence. So they have gaps, but we do have a number of fossils evidence. And also some other evidences can come from homology, which means basically um, homologous structures, which is a microevolution, which is from a divergent evolution. So there are some examples like you see here, homologous structures of analytical science. We can see between different species. Pinches of Galapagos are also good examples of homologous. And molecular biology is another evidence. Biogeography is another evidence. Fossils is another evidence. Homologies, direct observations, these are all evidences. And evolutionary trees are based on these homologous structures like cladograms. You can see here phylogenetic trees. And then this talks about natural selection, which is one of the evolutionary mechanisms. There are other evolutionary mechanisms, basically mutation. Obviously, we've seen this in many places. Also, how this works on your populations, and some other mechanisms, including genetic drift. You can see here genetic drift, gene flow. These are all the mechanisms under evolution. Okay, so under the genetic drift, also make sure you look into bottleneck effect, and specifically like how bottleneck effect works. So, and also founder effect, how bird cross the river to the other side of the river, and basically they can actually start a new population. So this is chapter 11. Chapter 12 basically continues this, but as a macroevolution. Chapter 12, what's unique about that? So make sure you look up carbon dating, like has 5,730 years that can be used as like finding the age of a fossil record. So remember, chapter 12 has everything chapter 11, but also talks about macro level evolution, which is about species. So it also talks about examples like bacterial resistance and barriers and allopathic speciation or sympatric speciation. So a few key points here. So The phylogenetic trees are also here, so make sure you look up the phylogenetic trees. So, for example, how squirrels branch out from the mammals. So we talk about big changes between species, not like previous chapter, but there are barriers like pre-zygotic or post-zygotic. There are some examples here. And then how universality of genetic code can help here, and then fossil records again, and geological evidence. So make sure you study all, and then make sure you actually do some reading from the Orange Book, the chapter 12. And to recap, so we have 3 chapters, 10, 11, 12, DNA technology, and then 2 evolutionary chapters. So keep studying, keep reading, and if you have questions, just send me an inbox. So feel free to comment here, and thank you for watching and happy studying.